Welcome to our Unlocking New Horizons session on clinical trials. I'm pleased to introduce our two speakers today. We have Dr. Saman Maleki as an associate professor at the Departments of Pathology and Laboratory Medicine and Oncology at Western University, and a translational immuno-oncology scientist at the London Health Sciences Research Institute and the Ontario Institute for Cancer Research in Canada. Dr. Maleki has led the development of multiple phase 1 and 2 trials in advanced pancreatic cancer, melanoma, renal cell carcinoma, and lung cancer. And as his co-panelist, we have Dr. Charlene Gill, who is a medical oncologist specializing in gastrointestinal malignancies at BC Cancer, Vancouver. Following the completion of her MD, she completed residencies in internal medicine, medical oncology, subsequently completing a fellowship in GI oncology at the Mayo Clinic, as well as a master's of public health from the Harvard School of Public Health. She is presently professor of medicine at the University of British Columbia, and her areas of clinical expertise are in colorectal, pancreatic, and hepatobiliary cancers. So please give a warm welcome to both of our speakers for this session.
Hi, good morning. Can you hear me? Great. Okay. Would you like me to share my slides?
Whatever you prefer.
Okay, great. I think Dr. Maleki and I had sort of planned it that I would maybe start and give my perspective as a clinical trialist, and then we'll kind of lean into sort of the more translational opportunities that exist across Canada. My regrets for not being able to be there in person, and I hope that this goes off without a technical glitch, but can I get confirmation if you can see my slides? Yeah. Yes. Okay. Okay, amazing. Thank you. So, as you heard, I'm a GI medical oncologist. I'm based out of BC Cancer in Vancouver, and I also have the privilege of chairing the disease site for GI cancers in the Canadian Cancer Trials Group, which is our national cooperative clinical trials mechanism. I was asked to kind of speak to you today about the clinical trials landscape and really with a focus on what is happening in Canada. I'm sorry, I'm trying to turn my phone off, but and so what I thought might be helpful, I know this is an audience with different levels of expertise, including a lived experience, was to maybe start very high level and talk about sort of, what are some of the constructs and the paradigms around, clinical trial design and conduct, not only in Canada, these are fairly internationally accepted sort of principles. And I think it starts with a fundamental understanding that when we're asking patients to consider a clinical trial, what we're asking is to engage in what is essentially a scientific experiment. And typically for the evaluation of a therapy that we hope we can demonstrate has both statistically significant and clinically meaningful benefits, sufficient that it will, change practice in a positive way for us to help our patients. And why are clinical trials important? I mean, I may be preaching to the converted here, but we know that in addition to the sort of goal of informing and improving and changing practice, medicine is very much an evidence-based paradigm. So we and often the best questions really come from our experiences with patients. But we cannot rely on that kind of anecdotal experience as evidence sufficient to change practice in a way that we are reasonably confident this is the risk benefit ratio is in favor of benefit for patients. So it's about how do we translate that scientific inquiry into clinical practice. And it is also about, and we're going to hear about this, I think, through Dr. Maleki's work as well, is about how do we really, from the bench to the bedside, translate those advances that are being made in basic science and scientific research into something that affects clinical care at the front line. Um, when we talk about clinical trials and how that the levels of evidence required to change practice, this is still the conventional paradigm that is used, and we define trials by the objective, and in the development of a potential new therapy for patients. So a phase one trial. Historically, these have been called sort of first in human studies, but these are basically studies where typically the endpoint is to determine a safety profile, determine sort of what is the best dose for considering this potential new treatment. And obviously there is a goal to try to look for some early efficacy signal, but that is not often the primary goal of a phase one trial. The phase two trial is where you've done the dose finding, you kind of have an understanding of the safety, is where you're trying to get a sense of, is the signal of efficacy sufficient? We call it, so the go no go, where this can move on to a phase three trial, which is the conventional sort of anchor of a level one evidence, typically a randomized trial against the standard of care practice, where it's powered in a way that you want to be able to demonstrate that if there is a benefit that is seen with the new intervention, it's real, it's a true, statistically a true benefit. So those are the trials that change practice. And then phase four trials are sometimes referred to as sort of once a new therapy is available, really trying to kind of expanded safety data and sort of in that implementation. Um, Patients' rights being protected in a clinical trial are paramount. So that is really anchored on the principle of informed consent. And there are very strict operating guidance around what constitutes informed consent. There's tri-council policy statements, and everyone has to be really involved in the conduct of clinical trials, has to be trained appropriately to understand informed consent. There's scientific peer review that's done for approval of clinical trials. We operate in institutions that have research ethics boards that ensure that those protections are in place. And then there are often, particularly for phase three trials, independent data safety monitoring boards that are evaluating the progress and safety of a trial, and obviously independent because they have no vested interest in the outcome. This is really to ensure that patients' safety and rights are protected. Um, we know that obviously, even when a trial has enough of a burden of evidence to get to a phase three trial, that all trials are not positive practice changing trials. And so there's lots of considerations in planning a trial that we hope, ultimately will help improve lives of people living with cancer in the cancer clinical trials realm. And so what makes a good clinical trial is often it's answering a relevant question that will change practice. And so what that means is that for me as a someone who's treating my patients and also offering them a trial, I have to have what's called a state of clinical equipoise. I have to have um, genuine, uncertainty that this experimental treatment will, may have uh, um, may be just as good as the control arm. So what I mean by that is sometimes, I'm sure you've all seen this, there may be sometimes there's some very early data, very limited numbers of patients, really promising signal, and suddenly everyone's like, well, why would we not offer this to all our patients? And we say we don't have the level of evidence. In order for that trial to be done, I have to be reasonably confident that I'm not offering my patient an inferior option in the standard of care arm. There has to be Everyone has to be on the same page in terms of what is, of the stakeholders involved in terms of the intent of the trial, the goals, the endpoints. So this is the academic side, industry side, when trials are done with obviously our industry partners, regulatory sides, Health Canada, and that supervision, and obviously our patients. And so, and that often takes a lot of planning. It is not unusual sometimes for it to take more than a year, if not longer, for trials, even when you have the preliminary data to meet the rigor to get to the point of a trial that will be a phase three trial that will be offered to patients. And when we say successful trial and a trial that changed practice, sometimes it's also really important to recognize that trials that uh, demonstrate absence of benefit or no benefit are also important. So we're not exposing patients to treatments that ultimately are not helping them live longer or live better. There are challenges to this kind of design. So it's rigorous, as I hope you're kind of seeing here, but and I've also said it's lengthy. And so for my patients, and I'm sure many in the audience have is experience, we don't have the luxury of waiting years for new treatment. So this is a lengthy drug development timeline, and his traditionally, and this is where there's a lot of interest in trying to really have adaptive trial designs, is that you're testing one, a treatment at a time. We also know that cancer is rapidly changing. Our understanding of subtypes of cancer is changing, and we're going to hear a little bit more about that. And so, the kinds of eligibility criteria within trials sometimes can be constrained by older definitions and may not reflect the rapidly changing landscape. And so, novel and adaptive, trials designs are required, and that is recognized. I wanted to take a few minutes to talk about the CCTG. I mentioned I chair the GI disease site for the Canadian Cancer Trials Group because that is really the the cooperative group mechanism. So when I say cooperative group, I mean a non-industry support. So this is the national network. The Canadian Cancer Trials Group is an academic cooperative oncology group. There's a network of basic science researchers, scientists, patients, investigators, clinical investigators like myself that design trials across the spectrum of the cancer care continuum. So from prevention, treatment, supportive care, survivorship. And, the hashtag for CCTG is trials that matter. So really it's about trying to answer those questions that sometimes can only be answered through this kind of a cooperative group mechanism. The Canadian Cancer Trials Group is primarily supported by the Canadian Cancer Society. So a lot of the work that's being done through the CCTG is through philanthropic donations across Canada, and many of the trials are grant supported, so grant funds through the CIHR, the Canadian Institute for Health Research, and we are also part of the North American or the National and Clinical Trials Network, which is the North American consortium of collaborative groups. So with the U.S. And that's referred to as the NCTN, and our participation there is also supported by grant funding. There are more than 85 CCTG member sites across Canada, and these are listed here. So these are all institutions that have been accredited, if you will, to conduct cancer clinical trials through the CCTG, and they receive sort of that oversight and support. The CCTG is structured with a number of scientific committees. So the GI pancreatic cancer sits within the GI disease site, which is part of the GI scientific committee. Then there are oversight committees. So we have a data safety monitoring committee, a clinical trials committee that ultimately approves trials that are presented through the network, a strategic advisory committee, and then, these are very cross-cutting and support committees. The GI disease site is co-chaired by myself and Dr. Chris O'Callaghan, who's a senior investigator with it, primarily based in Kingston, CCTG. Within the GI disease site, we have what are called disease-oriented groups, and pancreas is one of our 6 disease-oriented groups across the GI disease site. Because of our relationship with the NCI through that NCTN US network, we also, we sit in the task forces within the US, really trying to think about opportunities to bring trials for patients in Canada. And our pancreatic disease-oriented group is chaired by Dr. George Zagopoulos from McGill and Dr. Dan Runoff from BC Cancer. And this is kind of just to illustrate, obviously it takes multiple stakeholders and active engagement with patients. And, often a call out to Craig's cause, one of the pivotal trials that did change practice in pancreatic cancer was the trial of adjuvant FOLFIRINOX. And that trial, the participation in Canada was supported through, fundraising dollars raised by Craig's cause. So this is like, from the grassroots level to like, national bodies effort to bring clinical trials for our patients in Canada. I wanted to kind of end by highlighting current pancreatic cancer trials that are being conducted through the CCTG and what's up and coming. So this trial is a trial that's actually we're participating through the NCTN. So it's led by Alliance, which is a cooperative group in the US, and is asking the question about when patients have early stage resectable pancreatic cancer. um, Typically, the current paradigm or the standard arm, which is sort of that row on the bottom, is for patients to go directly to surgery and then receive adjuvant postoperative chemotherapy to reduce their risk of recurrence and try to eradicate any microscopic residual disease. And the trial that I mentioned earlier that Craig's Cause supported for Canadian participation established this as the standard of care, surgery followed by FOLFIRINOX. The question that's being asked is because we know that the risk of recurrence is very high in pancreatic cancer, and that pancreatic cancers do have a risk of early metastatic spread, those sort of microscopic disease disseminating early. What is the benefit of giving chemo before surgery? So what's called neoadjuvant treatment. And many studies have led to this phase 3 trial that single arm centers and institutions that demonstrated the safety of this approach and suggested that, yeah, maybe this is something that actually could help people do better in the long run, but we needed this level 1 evidence in a phase 3 randomized trial. So the experimental question is splitting the chemo, giving half of it before surgery, then patients going to surgery, and half after surgery. And the primary endpoint is looking at survival. This trial, I'm pleased to say, actually just this week announced that it's very close to completing its target accrual. So patients who are in screening will still be enrolled, but the hope is that this will really inform better how we can reduce risk of recurrence and improve survival for patients with resected pancreatic cancer. The other program that's being run through that's being supported through CCTG and through the CCTG sort of academic network and Marathon of Hope, which I'm going to talk about in a moment, is called Maestro. So Maestro is a genome sequencing to inform novel treatments in pancreatic cancer. And so we talked about novel and adaptive designs. I mentioned that earlier. So this is kind of trying to create a platform where multiple interventions could be tested based on molecular profiling of a patient's cancer. And so the first experimental question that's being asked is in the proportion of patients with pancreatic cancer who have KRAS wild type disease. So we know that, and we're going to hear about this a little bit more, but we know that RAS mutations are very common in pancreatic cancer, but there are a proportion of patients whose pancreatic cancer does not have a RAS or KRAS mutation. And in other cancers, there's been benefits that those patients their tumors may respond to an EGFR inhibitor. So the cohort that's ready to be open soon is looking at KRAS wild type advanced pancreatic ductal adenocarcinoma receiving FOLFIRINOX and panitumumab. And this is a collaboration where the sequencing is being supported through the Marathon of Hope. The novel drugs that are going to be tested in different molecular subtypes informed by that sequence are going to be supported by partnership with industry. And the hope is that this can allow for some rapid iteration to look for signals that can allow for new treatments to be expedited more quickly. And then I wanted to mention the Terry Fox Research Institute Marathon of Hope. I think as a Canadian, I cannot think of a figure bigger than Terry Fox in terms of his message to really bring together advances in cancer. So the Marathon of Hope network is a network across Canada, and the Pancreatic Cancer Working Group has been very active, and really a large part of the focus has been on advancing molecular sequencing, whole genome sequencing through Marathon of Hope funding. And in the recent budget, in the recent national budget, the Canadian budget from reported last month, thankfully there has been funding allocated to continue to support the grant funding for Marathon of Hope. So that's very positive news for us. And then lastly, I wanted to kind of highlight this work. We're talking about molecular profiling just because it is close to home. So this is work that's led by Dr. Jennifer Knox at the UHN. And one of the other questions that often comes up in the treatment of patients with advanced pancreatic cancer is how do we, we have these two options. Basically, we have FOLFIRINOX and we have gem-nab-paclitaxel based from an earlier study called IMPACT. And they've never been compared head to head. And also there is some, are there some patients whose tumor biology may actually respond better to gem-nab-paclitaxel or FOLFIRINOX or vice versa? And so through two trials, the PASS-01 was a randomized trial. One of the learnings that has come from Dr. Knox and her group's work at UHN is that there is a molecular subtype classification, classical cell versus basal-like cell. And basal-like cell seems to be more sensitive to gemcitabine-based treatment and potentially more resistant to FOLFIRINOX. And could this kind of allocation, understanding upfront, help us better assign treatment for our patients based on that? And I know this work is still ongoing, but just kind of a view as to from a precision oncology lens where things may be going. And then finally, I think this was just very recently launched as a resource to enhance the ability to find and access clinical trials in Canada. So I've talked about the cooperative group, mechanism, but we know that a number of centers across Canada have not only CCTG trials or trials with other cooperative groups, but also a number of industry-sponsored trials. And this website it's a searchable website, and it's for all cancers. But you can search pancreatic cancer, and it will list for you, the trials that are open across Canada for different indications and how to contact. So I think some of us are familiar with the clinicaltrials.gov, which is a kind of an international registry of clinical trials, but this really brings it home more to Canada. And this was an effort led by the Canadian Cancer Society, Cancer Trials Canada, and QCROC in Quebec. So this is a helpful resource. And then that was kind of my last slide. I'll hand off to Dr. Malecki. Thank you. Maybe I'll stop sharing.
Thank you very much, Dr. G. That was fantastic. Thanks to the organizers for inviting me. I gotta say, my main disclosure is that I'm a tumor immunologist and a translational scientist, and in a room full of practitioners, it feels like I should be here getting lessons from everybody here. So. But let's jump in. So pancreatic cancer is it's a really difficult disease to combat, and we know that. And now that the 5-year survival has improved to 13%, it's obviously that's progress, but that's really shows us how much work we still have to do, and we still have only surgery as a curative option for this treatment. And one of the biggest issues with pancreatic cancer is that although it's only about 3% of all cancers that are diagnosed, but it's about 80% death rate. So, and that makes it a very lethal cancer, which is also on the rise in North America and elsewhere. And that gives us pause really to think about what should we do about a cancer that is so lethal. And as Dr. Jill mentioned, we really have two options to treat these patients. And as everybody in this room knows, most of the treatments that we have, most of the systemic therapies that we have are really suboptimal. They're not really as effective. Obviously, they are helping patients as much as possible, but there is a lot of work that needs to be done. But that also, I guess, summarizes the state of pancreatic cancer. In pharma, there's an expression, they say pancreatic cancer is where all the drugs go to die. And that summarizes why there hasn't been a lot of progress in pancreatic cancer, because a lot of times you see that the drugs that are being even discussed this morning about targeted therapies and things that people are trying to use, using more personalized approaches that haven't been really developed for pancreatic cancer. They're developed for other cancers, and now we're trying to find the same mutations and apply to pancreatic cancer, meaning that we still have a lot to do. So if it's really for us to do something it does require a lot of good science, a lot of good effort, and probably also a lot of investment to bring to this field to make changes. I'm going to highlight some novel treatments that are on the rise. And obviously, KRAS targeting agents are offering some exciting targeted therapies. But obviously, this is a much smaller subset of patients. But there are some first generation of these drugs that have shown some good objective response rates and with some reasonable PFS as a targeted agent, like sotorasib and adagrasib, and they're in the clinical trials. None of them are really approved in Canada yet, and that also tells us that we need to do more to get our patients access to clinical trials and hopefully access to some of these targeted agents. The pan-RAS inhibitors are more exciting in many ways because KRAS, as you know, it's a very common mutation in pancreatic cancer, and hopefully these drugs that are now the phase 3 trials are ongoing, they can probably be very important for this disease site. And also the KRAS G12D and also G12V and other mutations that they are now being targeted with these drugs. And as I mentioned, unfortunately, there's no drugs in Canada approved yet, and we are kind of behind in terms of getting access for these drugs. And hoping that Health Canada and Cancer Care Ontario and other provinces, we can work with our partners to effectively get patients access to these drugs, but mostly probably through clinical trials for some time. But I'm going to highlight some of the emerging treatments and things that are more near and dear to my own heart. And that's about some immunotherapies. As you know, as full disclosure, immunotherapy doesn't work in pancreatic cancer, okay? So everybody knows that unless you're an MSI high patient, immunotherapy is not going to work on you. But what's really interesting is that there are some studies that people have looked at pancreatic cancer patients who are long-term survivors. These are patients who have the same mutation profile as everybody else. But when you're looking at them, and by that I mean the driver mutations, like KRAS mutations, but when you're looking at them, you see that the median survival is much higher in these subset of patients than what's a typical pancreatic cancer patient. And when you're looking at the baseline tumor of these patients, this is a study that was published in Nature in 2017 by Vinod Balachandran from MSK. They showed that there was a high level of T-cell infiltration into the tumors of these patients. And interestingly enough, these T cells also showed an activated profile. So that means there was a productive anti-tumor immune response in these tumors. And that's not surprising to see that these patients are doing well, because there are other cancers like ovarian cancer, for example. That's a type of cancer typically doesn't respond to immunotherapy. But if you have a high T-cell infiltration in the tumor, those patients do really well. So what's also really interesting in immunophenotyping of these tumors is that you also see increased dendritic cells and some immune pathways activated in those tumors. As I mentioned, this is showing that you have a productive cancer immunity cycle. And that's really important to consider, because this is telling us today the immunotherapy agents we are testing in the patients, like checkpoint inhibitors, they're not working. But naturally, there are some tumors that are responding really well to anti-tumor immune response in the context of standard of care, which is those two chemotherapy options that was mentioned, FOLFIRINOX and gemabraxine. So when looking at these tumors in a more detailed way, what turns out to be driving this T-cell response, and I know this is a busy scientific slide, but what's really shown here is that there are some neoantigens that are enriched in the tumors that they respond really well. In fact, in this figure here, what you're seeing, that the red dots are neoantigens, and what we can see is that they can fall below the line here. That means the tumors are losing those in the patients that they don't respond, or they actually are short-term responders to chemotherapy, when they don't have enough of these neoantigens. Or what you also can see is that when you're looking at metastatic sites, these mutations are often they are lost. That means there are some neoantigens that they exist in the pancreatic tumor. As the tumor goes through the evolution, it looks like it loses those mutations. The T-cell response is gone. The patient's not responding really well. But and this is actually it's an interesting case here, we had somebody contacted us through the Baker Center. There was a patient that wanted to donate their body to pancreatic cancer. Some of the people in the room are aware of that. So this is actually a study that they did on a rapid autopsy of a patient's body, and effectively they looked at the loss of new antigens. So but that's something that we also got called here about as a patient wanted to donate their body. So here, what you see here is that the investigators looked at the peripheral blood in patients who are long-term survivors and predicted the new antigens from the baseline tumor of those patients from the biopsy. And what they saw is that years later, you can still mount an active immune response in those patients who are living 7-10 years later. That means active immune cells are circulating throughout the patient's body, still recognizing mutations. And what's also remarkable is that some of these antigens are actually microbial antigens. They're not necessarily tumor antigens. And what do I mean by that? It seems like there are some microorganisms that might be colonizing the tumor, and the immune system is recognizing those antigens and mounting a response to it that as a side effect controlling the tumor. And we'll get to this in the next set of slides that I'll talk about. And this part here is also showing that the patients who are long-term survivors, they're sharing those antigens. That's actually a microbial antigen that's shared with the tumor. So this slide was shown earlier today. This is a trial that was run by the same group that made lots of headways actually when it came out initially in New York Times and other popular media. And what they did was this was a collaboration between MSK and one of the mRNA vaccine companies. And what they did was they did a personalized analysis of the tumor's antigens. And then they sequenced the tumors and they predicted the new antigens, okay? And then what they did was they created an mRNA vaccine based on those new antigens. And then they gave it to the patients through a prime and boost cycle. Okay, the patients received one dose of atezo, which is an anti-PD-L1 treatment, 6 weeks post surgery, and then they got the vaccination, and then 21 weeks later, they got a modified regimen of FOLFIRINOX. So just a side note, because some people think that chemo should destroy the T cells, but as you can see here, if you space it out, this is for trialists in the room, and if you're interested in designing a trial that involves the immune system, you can still give the patient chemo, but you want to time it a little further so that you don't have a lot of effector cells that they're proliferating that the chemo can kill, because at this stage, you have a lot of memory cells that are not really affected by chemo. So and what the results showed was this is obviously a very small study, only 8 patients in each cohort, but they showed that the patients who were able to mount an immune response against the vaccine, they were able to actually show a much better disease-free or recurrence-free survival compared to the patients who did not mount an immune response, okay? So, and this is now being tested in a much larger phase 2 trial. But that tells us that from the first study the group did, found that there were antigens or new antigen-specific T cells that seem to be driving response in patients, long-term survivors. And the second study is a rational progression of the first study. So we can probably now mount a more specific immune response in a more personalized manner for these pancreatic cancer patients and hopefully drive response. Obviously, everybody's excited to see what happens with this trial. So one thing that they showed in the same paper is that, as you can see, you're driving some of these new antigen-specific clones of T cells, and they actually last quite a bit in the patient's body. Actually, you can see that they last, some of them 3 to 4 years in the circulation, and they can detect these T cells. But if a patient does not mount that T cell response, they won't respond to treatment, and they become a short-term survivor. So another study that is also very interesting was a study that was published by Florencia McAllister's group from MD Anderson in Cell now about a decade ago. And what they saw was they also looked at the long-term and short-term survivors. And what they saw was that there is a group of long-term survivors that when they're looking at these patients, they do not seem to have a very different, any difference in terms of driver mutations in their tumors, but they have actually what we call a tumor microbiome that is more diverse than the rest of the cohort. So what's the tumor microbiome? So the microbiome is all these bugs that are living in our body. They work like a pharmacy. They're producing all these metabolites that's helping the function of the rest of our body. And as it happens, actually a lot of tumors, specifically GI tumors, happen to have their own microbiome. And if you sample a tumor and you see the bugs in the tumor, and you see the bugs look like a healthy bug in the gut, those patients do much better. And this goes back to that bacterial antigens that we talked about. So what this group did was that they looked at the overlap between the stool microbiome and the microbiome in the patients. And what they saw was that there was about a quarter of what constituted the tumor microbiome was similar to the stool microbiome, okay? And that's actually really important because we can change the stool microbiome. We can give patients something called a FMT, a fecal transplant, and change it, and I'll talk about it in a second. So what they did here was that they went to the short-term responders, and they got stool from them and colonized animals. And as you can see, they were able actually to change the stool, the gut micro or tumor microbiome by doing FMT from the short-term survivors into the mice, proving that you can change or colonize the tumor microbiome by doing a fecal transplant, okay? And what they showed that if you actually compare short-term survivors to long-term survivors and healthy individuals, you can actually drive an anti-tumor response and control tumor growth by just doing an FMT. And in this paper, they also show that this depends on CD8 positive T cells. Partially, T cells are recognizing these antigens that they are now presented in the tumor. And why is that important? Because we know that tumor microbiome, as we talked about it, it has this gamma protobacter bacteria that they live in the pancreas tumors. About 70% of pancreatic cancer patients actually have their tumors colonized by these bad bugs, and they are able to metabolize gemcitabine, in fact, and that's one of the resistance mechanisms to gemcitabine that we are giving these patients, okay? So knowing this data and our center being one of the effectively leaders in the world to do fecal transplants, we designed the trial with the help of John Lenahan and Daniel Hadi, who's a fellow here in the room, and he's going to have a poster. Make sure you go to talk to him about his work, and Mike Silverman and Dr. Simo Parvati. And this trial is called PDAC Chase. This is the first in the world trial in the advanced or unresectable patients that we are giving them FMT. So the patients come in, they actually go through a bowel prep. So we want to make sure that the gut microbiome is cleansed. And then we give them one full dose of our oral FMT product. It's called London 101. And then we allow for 1 week of engraftment. So we want to make sure that the microbiome actually has time to engraft. And then we also collect samples from different time points from these patients, and then they are going to get their gemcitabine treatment, okay? So, and then we follow the patients through samples and the scans as usual. So a lot of people in this room who are part of the Baker Center have contributed to these trials and putting patients on this trial for us, which we are very grateful. So this is just some data from the first 10 patients in the cohort. As you can see, this is the older population of patients. So the patients who John and other oncologists in the room would probably just treat with gemcitabine. They have all sorts of different KRAS mutations. And as you can see, they're advanced, effectively stage 4 patients here with different sites of metastases. And in terms of toxicity, because the primary endpoint of this trial is toxicity, we didn't have any unexpected or previously not reported toxicity. In fact, it's mostly what you expect from chemotherapy in these patients, but everything from what I hear has been manageable so far. So in terms of response, this is the early look at the data. This is very preliminary, but what you can see in the first 10 patients, we had 3 patients who had a partial response, and effectively everybody else showed a good stable disease to the treatment. And this was also mirrored by a clever analysis that John did with CA19-9. And you can see there is a good biochemical response to the therapy. So we are very excited. We have 10 more patients to go, in fact, 8, because I think Daniel put 12 patients so far on the trial. So that's one area that we are trying to advance the treatment. Obviously, our goal is to finish that trial. And if the data is promising, then I'm going to go beg a drug company to see if they would give me immunotherapy to add to this regimen. And so we can do chemoimmunotherapy with FMT. So this is an example of trying to develop CAR T therapy. I was at Society for Immunotherapy of Cancer annual meeting 2 weeks ago in Washington, D.C., and at a cocktail party, I heard some folks from some of these CAR T companies talking about some of these CAR T products that are coming for pancreatic cancer. So they don't tell you much, but it was very interesting, the conversations, and might be something in the work. But this is an example of a Claudin 18.2 target. This is an antigen that's often overexpressed in a lot of cancers, but also in pancreatic cancer. And these are CAR Ts. You're getting blood from the patients, and then you engineer these CAR products into the T cells, and then do lymphodepletion in the patients and reinfuse these CAR Ts into the patients, back in the patients, okay? And as you can see, as the patients get the CAR Ts, they shoot up some of these C-reactive proteins, the body temperature goes up, IL-6 level goes up, interferon gamma. This is what we call a cytokine storm, okay? It's much, much higher than what patients experience during COVID. And then they often just get tocilizumab, and the pioneer of CAR T therapy, by the way, is Dr. Carl June, who's going to be in London on Tuesday, accepting the Taylor Prize, and I hope that people can go to that talk. So, but for this study, as you can see, this is an example of a patient with pancreatic cancer who was infused with CAR Ts. And as you can see, the patient actually mounted a very good response that lasted about 250 days in these patients. Unfortunately, after 240 days, the tumor came back with and lost its claudin. So that means the T cells put enough pressure on the tumor, evolutionary pressure, that the tumor has lost the claudin. And we can see the CA19 was stably down over time. But that's another example of how we can advance more immunotherapy in a difficult solid tumor area. So being in Canada, as you know, Dr. John Bell and some of the folks in Ottawa Hospital, they're the pioneers of oncolytic virus therapies. And there are some really interesting oncolytic viral therapies that are in the work right now, including the ones that we call them ARM oncolytic viruses. So these are viruses that you're effectively putting a gene, a transgene in them, and they're able to bring this payload to the patient's tumor. And then what they do is that they will express those molecules that would potentially affect the tumor environment. And it's really interesting, the science of this, because if you think about it, what does a virus like to go? A virus likes to go somewhere that you don't have an active immune response, like interferon signaling. You don't want to call an immune system to come clear you, okay? And maybe the cells are proliferating fast. Who does that? Tumors, okay? They don't want to call up the immune system. That's why viruses have natural tropism through tumors. And that's why the oncolytic viruses have this opportunity to get into the tumors. And this one called LOAD703, it's an adenovirus-based oncolytic virus. As you can see, it has a CD40 ligand and a 41BB ligand. And what it does, this one activates dendritic cells, and this one primes T cells. So if you remember from the earlier slides I mentioned, in those pancreatic cancer patients who were long-term survivors, they seem to have a both T cell response and the DC response. And that's what you can effectively engineer here. And they have shown some initial preliminary good results with 44% response rate and a very good disease control rate in these trials. And there's some other vectors that are being developed, obviously, like the VCNO1, that actually has shown some promising OS improvement. And there are other ones based on different disease, different oncolytic viruses that are in the work. So this is to say that we should not despair when we're dealing with a disease like pancreatic cancer. We need to do a lot of good science and a lot of investment in a very difficult field to hopefully advance the advance the field and help patients. And hopefully with the help of foundations like KRAS and others, we can effectively advance science. With that, thank you very much. Happy to take questions.
That was a really great overview there, Saman.
Thank you.
Appreciate this. For people who don't know me, I'm Chris Pinn. I'm a lead researcher at the Baker Center. And I actually have two questions. The first one is kind of to use directly, and then the second one to both of you, just as an opinion. So you talk about, I mean, you hit upon about 1,000 different studies, it seemed like. And one of the things that came to me is that all of these are based on like observations in an individual or group of individuals, and you see a lot of variability when you target something. In one case, you're targeting mRNA vaccines. It's obviously to something specific. And then the second one you're talking about was Claudin 18 being targeted.
Yeah.
And does this mean that if we really want to do this, first of all, it seems like obviously multiple approaches, but do we really want to be looking at individual tumors and personalizing these approaches? Because I think especially with when you talked about CAR T cells and vaccines, that's a specific target you're looking at. And to think about that as something that's going to hit many of patients seems unreasonable. But what do you think?
That's absolutely true. So there's two type of studies I highlighted. One was, as you mentioned, mRNA vaccines and the CAR Ts. And for those studies, you definitely need to make it individuals, which is expensive, difficult. But obviously, as technology is progressing, they're making headways in the labs. But again, it's going to take quite a bit of time. But with the heterogeneity in pancreatic cancer is more difficult. On the other hand, I talked about the study we are doing with the fecal transplant, that we are actually trying to also more optimize it with donor patient matching, but that's a more general approach when you can potentially combine it with something like the checkpoint inhibitors. I do think it's difficult to get away from personalization or personalized medicine in pancreatic cancer, but as I'm aware of the efforts that you and others are pioneering in our center, like using the organoids, for example, so that you have a screening tool that you can potentially, the patient comes in, you can screen it and say this patient would fit for this type of treatment, potentially. So that's an area that I think from a bench-based scientist we need to work on to develop these tools or screening tools initially to effectively triage patients to what would be potentially an ideal treatment for them. I think that's going to be the future, hopefully.
The second question I have is more general, and it's really about clinical trials. And and over the last 30 years that I've been in science, you see many more failed clinical trials than successful ones. And you think about how we set these up. And I know, and this gets now into maybe the minutiae of trying to get an REB and et cetera. But I feel like we develop these trials based on principles from years ago and what we can and cannot take. And I'm wondering if we're at a situation now where we should be thinking about all of the corollaries that we pull in, all of the extra stuff. Like we can get RNA, we can get tissue, we can get DNA, we can get all these things and do this analysis. And that can be informative, especially when something goes wrong. But I feel like we're hampered by our ability to get all of that information. And maybe that's part of the REB process, and maybe we need to standardize this across the country to be able to say, here are all these studies being done. How do we get all this other information that when it goes wrong, we can now find out maybe there's one or two patients that responded. What is it specifically about them as opposed to just something spurious? I'd like to get your opinion, Dr.
Yeah, I'm going to take the first stab at this, and then I'm going to let Dr. Jill to talk about it. At the Society for Immunotherapy Cancer, SITC is now brokering a deal with a bunch of pharma companies to see if they can get access to control arm of trials, data from patients, because pharma collects quite a bit of rich data from patients on the trials. And the question here is that can we get access to the patients who are on the control arm? This is standard care arm, really, to combine all of that data together and learn something from it. And believe it or not, that has been very difficult too. So I guess that's one of the areas that maybe something like the cooperative group or CCTG, and Dr. Jill knows this better than me, can come in and broker a deal that people who are contributing to this data goes to a center and they can share the data. And something like maybe Baker Center can do that if we have trials that are run through Baker Center and everybody who comes in can get access to the data and share it together. Dr. Jill, I think you have better response than me.
Yeah, I'm not sure I have a better response, but it's a really important question. I think, we talk about delayed progress in clinical trials in cancer, and pancreatic cancer, unfortunately, is definitely the paradigm where it's been very difficult to generate a successful positive clinical trial that has changed practice. There are maybe 4 in the last 15 years that have truly changed practice. And so what happened, I'm old enough to remember that there was a period of time where there were a number of like phase 3 trials that were negative, a huge investment of patient effort, dollars, research, capacity effort. And it was to a point where the NCI basically called a moratorium on clinical, like in pancreatic cancer. It was like a timeout. We need to rethink how we are conducting these trials using this traditional model of design. You go from phase 1, phase 2, you see a signal, you go on to phase 3. And so that led to a bit of a step back in clinical trial conduct, to be quite honest. And now we're seeing that resurgence again with a lot of the interest you've heard from Dr. Maleki, immune therapy, RAS becoming more and more potentially druggable as a target. And so how do we learn from that is say, well, how do we have to embed correlatives in trials that will allow us, positive or negative, to learn more about that disease state and what is the next important question. And so within CCTG, as many of you know, there is a embedded within each disease site, there is a correlative sciences tumor biology committee. To give an example, like in CCTG, we conducted the PA7 trial, which was a trial of immune checkpoint inhibitor combination, durvalumab, a PD-L1 with tremelimumab, a CTLA-4, in patients with advanced pancreatic cancer in combination with chemotherapy. And there was a lot, there was good reason to think that there would be something here. But unfortunately, that trial did not meet its survival endpoint, but it included ctDNA correlative analysis, and some of the data that has emerged from the trial has really informed what is the somatic mutation landscape in PDAC, both from the control arm and the experimental arm. What are the potential populations of patients that in fact may be more responsive to immune therapy? And there have been several publications since then. So you're right. It's not just, pardon me, a positive or negative trial. It really is trying to make sure that we are setting ourselves up in a way that we can get as much in 2025, as much correlative science information to inform what that next question should be. So not really an answer to your question, but I think it's recognized that this is an issue.
There's a question online.
It's Joan Miguel Romero. Do you want to unmute yourself, Joan?
Sure. Can you hear me?
Yes.
Hi, I'm John Miguel Romero. I'm an MD PhD student in Dr. Zagopoulos' lab. It's unfortunate I can't be there, but I'm glad that there's a Zoom opportunity. I had 2 questions. The first one was for Dr. Maleki. I'm curious what your opinions are on tumor mutation burden and whether the community has shifted away from thinking of TMB and neoantigens as a better predictor of response to immunotherapy. And then the second question is hitting on what Dr. Jill has said. I think sometimes when you look at the literature, there seems to be a disconnect between the basal and classical subtypes and immunotherapy subtypes. So I'm curious if you've looked at in your data, if there's any associations between the basal and classical subtypes and kind of these responses to any immune work that you're finding.
Okay, for the first question, I gotta say we have better data on TMB and lung cancer. Even the study that I mentioned, I showed about the neoantigens, what they refer to that was predictive of response and survival in those patients, what they define as high quality neoantigens, and those are the ones that the peptide would bind to the MHC groove much stronger and mount a T cell response. But that doesn't often come, or it's not really directly correlated with TMB. That's where the TMB falls short. That you don't necessarily, you may get more shots at the goal with high TMB, but doesn't mean you're getting those quality neoantigens that can be predictive. But still, a tool in a toolbox for a disease that's difficult to treat. And if a patient has a high TMB tumor or high tumor mutation burden, so there's a possibility that they have more productive or more high quality neoantigens. And the question is, do they have MSI high tumor at that point? And if that's the case, obviously they would qualify for immunotherapy. And that also goes back to what I mentioned to Dr. Pin here, that we need to develop better tools in the laboratory to model these questions and see if we can predict, based on a higher TMB model, better neoantigens to see if that then can be translated. One thing that I want to highlight here is that I tried to give this talk in a sense that you'll see there was a study that was done that put the rationale together, and then there was a trial that was launched. And that's in two examples, the microbiome trials with us and the neoantigen trials with the mRNA vaccines. And I think we need to do more of that in the context of translational research to have more productive, hopefully, answers for a problem like pancreatic cancer.
Okay, thank you. And then the second point of, have you looked at basal and classical with your data?
That's for Dr. Jill.
No, Dr.
That
Malek, it was more for you. It was highlighting the
was for
fact.
me.
Yeah, it was highlighting the fact that Dr. Jill showed that nice data from Compass and PASS-1, and I think there's the transcriptional subtypes, but I don't see that being often looked at in an immunotherapy or an immune context. So I'm curious if you've looked at basal versus classical.
No,
And
that's a great question. I think that's more looked at in triple negative breast cancer, and there is actually a better response in the I believe in the basal subunits in triple negative breast cancer to anti-PD-L1 therapy. But I don't know of any studies that people have looked at that in pancreatic cancer. But definitely an area of interest for immunotherapy research.
okay, thank
Yeah.
you.
And it's certainly maybe something that Dr. Knox's group is looking at. I don't know offhand, but you're right. Most of that comparison was really trying to answer the gemabraxane versus
Hmm.
FOLFIRINOX question, but it's yeah, it is a really important classifier to really see how we can apply that potentially to immune therapy would be interesting.
For sure. Thank you.
I have a non-scientific question, but a question from an advocacy standpoint. So we know that patients perform better when they're receiving PERT. So I'm just wondering, when clinical trials, come into hospitals and patients start participating, is there any thought that goes into, part of that eligibility or, putting them on PERT while they're receiving the clinical trial treatments as well, just to give them that? We have such a low enrollment anyways in clinical trials. So that was another question I have. But I'm just wondering if there's any thought that ever goes into making sure that their nutritional needs are all met, so that when they're facing clinical trial treatments, that their bodies are as strong as they should be, and have a better quality of life, and pair those two together. Because we know Dr. Keith Roberts and Ann Landers and Enrique Dominguez Moniz and Poppy Casvis, and the list goes on about, 14, 15, 16 years of research about the benefits of PERT almost being almost the same as chemotherapy, and that the patients who are receiving PERT correctly, a correct dosage, are living longer and have better quality of life during treatment, or people that couldn't receive treatment, then receive PERT, were then able to go into treatment in clinical trials. I'm just wondering if there's ever, consideration about a partnership between the clinical trials and their nutritional aspect.
Yeah, maybe I could take that question it's a great question. I think that, I mean, I think it speaks not only to clinical trials, but what represents best practice and standard of care. And you're right, we know that pancreatic enzyme replacement therapy in this population of patients can be very beneficial. So So the honest answer to your question is no. Specifically, the intent of a clinical trial when it's designed is that when patients are being offered standard of care treatment, we often say it's with complemented, especially in the advanced disease setting, with what constitutes best supportive care, right? Best symptom directed care, best symptom management care. PERT is still viewed more from a symptom management lens, even though I recognize what you're saying in terms of like you can't disentangle patient outcomes because if patients if their nutritional needs are addressed, they are better functional status and performance status, they may be more eligible for not only different standard of care treatments, but also for clinical trials. So there's some downstream consequences that are clearly logical. The one thing I will say, and this is is that oftentimes, just in general, as a rule, there is an observation that patients who participate in clinical trials, whether they're on the standard of care arm or the experimental arm, or whether the trial is positive or negative, have a better experience. And part of that is despite the increased intensity of being on a trial and some of the uncertainty around that. And part of it is because in a clinical trials model construct, I'll just give an example in my clinic. When a patient is on a trial, they have a dedicated clinical trials nurse who is monitoring them very closely. And what I often find is that, and not that our clinical nurses are not amazing, but when patients are in a clinical trial, there's a different level of intense monitoring. And so things like, an early change in weight or, something is going on, it gets flagged more quickly and sometimes reacted to more quickly. And so, what we want to like our goal is we should be offering the best available care for all our patients, whether they're on a trial or not. But patients are under more of a lens, when they're monitored more frequently, and sometimes that leads to things like patients being prescribed enzyme replacement therapy more commonly, and offered nutritional counseling and support. Although that's a kind of it's a recommendation for all patients with pancreatic cancer, but that may happen more often. So again, it's kind of more of a comment than a clear answer to your question, but I do think that when patients are on clinical trials, there is, there's a different kind of team behind them that's maybe catching these things more quickly or identifying it more quickly.
I just add two quick comments to that, like being cognizant of the time. One thing is that I'm aware of, we are fortunate here at Baker Center, patients who are coming in, even if they're not on the clinical trials, they're getting some nutritionist and dietitian support, which is, I think, very important. And the second point I would make is that we are collaborating with in a study with University of Pittsburgh, led by Dr. Diwakar Davar. He's a medical oncologist. And I can tell you that they have done the study. These are the patients who are getting immunotherapy, not necessarily pancreatic cancer patients, but they looked at the diet and the change in the microbiome. You can see a clear survival difference in that patient. It's a survival difference in patients who are getting that diet support, and we are contributing to data on that. And I think that's going to be very important also in a disease like pancreatic cancer. There are complexities with enzymes and other things that patients require to have. With that, thank you very much.